Hello, this is Eric Topol for Medscape, and I have a great privilege to have a one-on-one with my friend Paul Offit, and my go-to for vaccines. And this is kind of an important time for vaccines, so welcome, Paul.
Thank you, Eric.
We had convened recently to discuss vaccines, and then just last week there was a really important 9-hour meeting, VRBAC, vaccine-related biologics products for advisory committee. And certainly, Paul, you were one of the key players there. Can you give us your overall sense of this long meeting that was had an intense public interest?
Yeah, I think it was good that it was open to the public, because I think what the public got to see was a look behind the curtain. I mean, what we struggled with. I mean, typically vaccines one does large phase 3 trials, tens of thousands of people that get the vaccine or don't get the vaccine. The data are evaluated, submitted as a biological license application to the FDA for licensure. But that's not this process. This process is not going to be a licensure process. These aren't going to be licensed vaccines. They're essentially going to be permitted for use through an emergency use authorization, which is frankly no different than any investigational drug. Which is odd, because it's going to be given to most Americans. So to do this under sort of permission through EUA is odd. And I think from the public standpoint, when they hear emergency use authorization, they think hydroxychloroquine. I mean, a drug that was approved through EUA that didn't work to treat the disease, didn't work to prevent the disease. 10% of people that got it had, cardiac toxicities, or convalescent plasma, also a product which has never been shown to work. So the fear, and it's a reasonable fear, it's an understandable fear, that what the FDA has done up to this point regarding these emergency use authorization drugs is really not done its job. That they have not stood between pharmaceutical companies and the American public to make sure that we got products that are safe and effective. They haven't. And so therefore they're suspicious of this process. What you saw here, I thought, was an attempt to understand the differences between what the differences were going to be between doing it this way, these unlicensed products, and doing it the way we always do. Because the fact of the matter is, these are large prospective placebo-controlled trials. I mean, the Johnson & Johnson trial is a 60,000 person trial. Pfizer's 44,000. Moderna's 30,000. That's a typical size to do any biological license application. I mean, the HPV vaccine was a 30,000 person trial. Our vaccine, the rotavirus vaccine, was a 70,000 person trial.
Wow.
So, pneumococcal vaccine, 35,000. So the size of these trials is typical. So why not do it through a biological license application? Why not do it the way we've always done it? And the answer is several-fold. One, and most importantly, I think the FDA would never license a product that was tested for this short a period of time. I think that's the biggest thing. That we're not going to know anything really about not just long-term efficacy, but even relatively short-term efficacy. I think we'll know that there's efficacy for months, but not necessarily a year, obviously.
Right.
Whereas, the HPV trial, although 30,000 people, that was a 7-year study. Our rotavirus vaccine was a 4-year study. Usually it's several years. So this is unprecedented in terms of length of follow-up. And also, and arguably more importantly, safety. We'll have a couple months of safety data after the last dose, which would pick up something like, say, Guillain-Barré syndrome following flu vaccine, which we saw. But you do worry about, vaccine-induced immune pathology, and at least Stanley Perlin, who was on this conference call, is a coronavirus immunologist, and he wanted at least 4 months. So that's the struggle. On the other hand, 225,000 people are dead from this virus within a year. So as you move forward with these kinds of trials, you go from phase 1 to phase 2 to phase 3. You just try and reduce uncertainty. That's the goal. How much uncertainty do you need to reduce? Do we need to wait a year, for example, to see what the 1-year follow-up, knowing that during that time, hundreds of thousands of people may die? Should we wait 2 years? Which Bill Hazeltine actually recently said, why don't we wait 2 years and get all the data, as we would do for a typical vaccine? And the answer is because, with the rotavirus trial, it's a 4-year trial, 60 children died a year. That's a little different than this.
Yeah. So Bill Hazeltine was arguing during the advisory committee that should wait 2 years before.
No, it was just a recent media.
Media. Okay. Yeah. Because I didn't think he was weighing in.
He's not on.
Yeah, he's had some unusual things that he said when I've listened to his segments. But okay, so one of the reasons that an EUA is appropriate is, as you're putting in the context of this pandemic, which is unlike the other vaccines that you reviewed. But another is that it takes many months to prepare a product BLA application, right? With 10,000 pages. And, this is a much more expeditious route, right? What
Yeah,
are
yes,
there other reasons besides those two?
yeah. There's one other reason, and it was subtly stated on one of the slides that was presented by Jerry Weir from the FDA, which was regard to supervision regarding manufacturing. I mean, when the FDA licenses a product, they don't just license the product; they also license the process and they license the building. And that is a pretty rigorous process. I mean, I noticed that for our vaccine, the rotavirus vaccine, Rototech, that was a 1-year process. You have to validate everything. You have to validate the computers. You have to validate how you're cleaning out the vats. You have to validate how you're putting the cells on a monolayer, however you're making the product. You have to validate every step of that. And I think that in that slide that he showed, there was one word in there that made me think this is a little looser than normal, which is the word generally. It's going to be generally acceptable. I asked somebody from the FDA about this to explain this to me, because when I looked at his few slides, I thought, it doesn't look like it's any different in terms of what the FDA calls CMC, chemistry, manufacturing, control. That it looked like it was no different than normal, which didn't make sense to me because I couldn't imagine how it could be that fast with doing it the normal way. And so there are some subtle differences there. But again, I wish we wouldn't call it an EUA. I wish we had another name for it.
Well, yeah, now you actually, that's an important point you brought up, because there is this pathway, expanded access, which could be used. Now, what do you think about that?
No, I think that, and certainly Marion Gruber, when she was on that, I mean, she's at the FDA. I think she talked about that as another way to think about this, and I think that makes sense, because one of the fears she has, and I think we all have, is Pfizer had stated that if they show that their vaccine, meets a standard for efficacy, 50%, 60%, that they would then give the vaccine to the placebo group. That she made a point in a couple slides that she had that they are under no obligation to do that. That under an emergency use authorization, that you don't have to do that, because it's still, frankly, investigational. It's
Right.
approved as an investigational product, because the minute you do that, the minute you and that could extend to other placebo-controlled trials, then you've lost data. I mean, let's suppose this vaccine is 60% effective. There may be vaccines out there that are 80% effective, 90% effective. If you stop doing placebo-controlled trials, you're not going to know that. And so that's the tension in all this.
Now, you're bringing up, this is, I think, one of the most important points of all, which is once a company says their vaccine works and files for an EUA, then there's this, I spoke to the Pfizer group, Catherine Jensen and her colleagues, and their plan was, well, we'll go ahead and file for an EUA, and we don't necessarily have to cross over our placebo participants, at least early on, because we certainly want to follow them. But there's this ethical dilemma. On the one hand, you're claiming some signal of efficacy, and on the other, you're denying the participants access to that efficacy. So it's a very fine line, right?
Yes, there's not a clear answer to this. You could argue that, if you're a 70-year-old person and you were in the placebo group in the hopes that you were in the vaccine group, and now you find out that the vaccine's 60% effective, whatever, you can see how that person could reasonably say, I think I should get this vaccine. I'm at higher risk. If I get this virus, I have a 10% chance of dying. I want this vaccine.
Yeah, so there's monstrous efforts of having, up to 60,000 people in a trial. The follow-up can be basically lost, the safety access or window. And so it's like a domino effect. If one of these vaccines clicks and says, we've got it, and it looks like Pfizer and Moderna are kind of leading in time in that. But then there could be a rippling effect on all of the programs, right?
Yes, I think that, because ethically, can you give a placebo to somebody who's at risk? Someone, say, who's obese or has diabetes or is older. I hate using the word elderly. I think that's what happens when you get in your late 60s. It's harder to say the word. Our elders, the more mature adult, whatever. But
I'm with you, totally with you. Wait, so you think, well, clearly we're going to need lots of vaccines here. And as you alluded to, hopefully with quite incremental efficacy, even perhaps a second generation of vaccines of these 200 different candidates. But is it a problem that people will ultimately take multiple vaccines along the way?
First of all, I think the only way we can immunize the whole population is with multiple vaccines. Secondly, I think it's going to depend on the vaccine. So let's suppose you got a replication defective simian adenovirus or a replication defective human adenovirus, and then there was a different sort of replication defective virus that also looked much better, but it was also an adenovirus. There may be enough cross-reactivity where then you wouldn't benefit from that vaccine, from the second vaccine. But otherwise, no, I think that for the most part, you would be able to have crossover. I mean, look at shingles, for example. Zostavax was given to people. I got Zostavax. And then shingles, the Shingrix came along. It's a better vaccine, a significantly better vaccine. And you can now get that, even if you've gotten Zostavax.
Yeah, hopefully it would work the same. Now that brings up a topic that I think is really interesting that a lot of people don't appreciate outside of your expertise. And that is the concept of superhuman immune response with vaccines. So we've already seen now, some almost 30 well-documented reinfections around the world of SARS-CoV-2 with genomics to prove that the strain was different in the initial infection and the subsequent infection separated by months. And it wasn't the genomics of the virus evolving that was accounted for that it truly was a reinfection. Now, of course, that's a small group of 40 plus million infections. To have some 30 or whatever the number is documented is probably, of course, larger, but many don't have genomics. The point being is that some people don't have an adequate immune response. And we also know that through antibody studies after neutralizing antibody studies after infection. So there are some precedents, and maybe you can review that, whereby, and I think you already touched on one with Shingrix, the vaccine could actually perform better than the typical human response. Can you take us through that? Because that's a key concept that I like to be optimistic, and I'm hoping that the vaccines will be better than human responses. What do you think?
Well, there's certainly historical precedent for vaccines being better than natural infection. The HPV vaccine actually induces a higher neutralizing antibody response than does natural infection, significantly higher.
Right.
Tetanus, interestingly, if you're infected with tetanus, you actually need to be vaccinated because the quantity of the tetanus toxin, so-called tetanospasm, that causes symptoms is actually subimmunogenic. So you get symptoms without developing an immune response. Similarly, for children less than 2 years of age, by conjugating polysaccharides like the Hib, Haemophilus influenza type B polysaccharide or the pneumococcal polysaccharide to a protein, a harmless protein, you actually induce a T cell dependent B cell response that if you had just given the polysaccharide, which is basically what you're doing when you're getting naturally infected, doesn't happen. So actually those vaccines are better than natural infection. So if you as a 6-month-old got a Hib infection, you actually still would benefit from getting a vaccine. Same thing with pneumococcus. So there are examples of that. I think here where that might happen is that there were 2 papers in Science that looked at the capacity of this virus, SARS-CoV-2, to induce antibodies directed against interferon. In other words, it induces it induces you to have an immune response against your own immune system at some level. And that's why people were worried at some level about convalescent plasma as actually having those kinds of antibodies, which could be immune suppressive. So I think this is a weird virus. I think anybody who's been watching this for the past year should not make any predictions about what's about to happen because it's been so surprising. But I think you're right. I think we may be able to get a vaccine that is better than natural infection because when you're naturally infected with the virus, it reproduces itself 10 times, 100 times, 1,000 times. When you're infected when you're given a purified protein or you're given messenger RNA, which is just one protein of the virus, that doesn't happen. So we'll learn. I think we're going to learn as we go here. And I'm sure that some of this knowledge is going to be painful over the next couple of years. So,
do you think any of the different types of vaccines, whether it's genetic, protein, attenuated virus, viral vectors, lend themselves more to a more potent immune response, or is hard to know?
Right. Well, so technically, as a member of the FDA's Vaccine Advisory Committee, I have to sign a form that says that I never publicly expressed any preference for a vaccine. But I'll try and say it more generally.
Okay.
If you were older, for example, if you're over 65 and you look at the sort of Shingrix Zostavax experience, I mean, Shingrix has 2 powerful adjuvants. One is sort of based on this so-called QS21, a saponin, a soap, and the other is monophosphoryl lipid A. The Novavax product, actually, their purified protein product actually has one of those adjuvants. So you wonder the so-called QS21-like adjuvant, whether that would have induce a better immune response. Similarly, I think the Sanofi-GSK collaboration, that adjuvant is also a powerful adjuvant. It's a squalane-based adjuvant. They're so-called ASO3 adjuvant. So who knows? I mean, usually for older people who, as you get older, your immune system gets more and more senescent. So the sort of powerful adjuvants may make a difference as it did in Shingrix, but who knows? Let's see.
Yeah. Well, that's what we, when we're kind of in despair, we try to find the bright sides of things. And the fact that there are these notable examples of vaccine programs that turned out to be better than what humans can mount as an immune response it's really great to know that. I don't think that's actually out there in the medical community very much yet. Now, another topic we discussed previously, but an update, there are 2 points. One is how important, and I think this really brought this home. And today, Tony Fauci and Andrea Lerner and colleagues at JAMA published on this, that just because we're starting vaccines, we got a long ways to go. We still need to be wearing masks. And the point being there that, as you touched on earlier, that you could be an asymptomatic carrier and be vaccinated so you don't get infected, but you are harboring the virus in your nasal mucosa. Isn't that right?
Yeah, no, if you look, there are many reasons to believe that natural infection with SARS-CoV-2 does appear to protect against moderate to severe disease associated with reinfection. Although there have been cases, not shockingly, that people get a moderate to severe infection, they get better, then they get a second moderate to severe infection. That is not typical. So unlike, say, strep throats, which you get again and again and again unmodified, or gonorrhea, which you get again and again and again unmodified. Those are examples where natural infection does not appear to protect against disease associated with challenge. That's not true here, because what we're hearing is not that. What we're hearing typically is got an infection and then later found out I had sort of a mild form of the infection, was asymptomatically infected. That's the usual story. And that's what you'd expect from this kind of virus. Also, there were challenge studies, human challenge studies done in the early 90s with human coronaviruses that found that you did get protection for a year, and the protection was generally against moderate to severe disease. Good. That's what you want. You want, knowing that natural infection appears to protect, means that you can make a vaccine that can, in theory, induce an immune response similar to natural infection and therefore protect. So all good news. But I do think that what you say is exactly right. If let's say you get a vaccine that's 75% effective, which we'd all be happy with at protecting moderate to severe disease, that's still 1 out of 4 people who you don't know who that is,
right,
could still get moderate to severe disease. And also, I think the vaccine will not be as good at preventing asymptomatic shedding or mild disease, because that's what the animal model studies teach us. The animal model studies teach us you can prevent pneumonia, but you don't seem to prevent upper respiratory tract shedding.
Right, exactly.
So I think that's what you need to wear a mask, and that's what worries me the most. People ask me what keeps me up at night. That's what keeps me up at night. That there'll be a vaccine out there. It'll be oversold as being sort of this magical powder that we're about to sprinkle over the United States and make all this go away. And people are going to say, I'm tired of the masks. I'm tired of the social distancing. Throw it all away. Engage in high risk activity, and we take a step backward.
Yeah, well, I think that point is central, and you emphasized that before, but I wanted to get to it again because I think a lot of people think the vaccine comes and the masks go, and we're looking at likely all of 2021 with masks. If you just judge at the timeline where vaccines will ramp up and get to a large proportion of people. Now that brings me to, how do you promote the public trust? You've spent your career on vaccines and dealt with, the resistance, the anti-vaxxers, venom, all kinds of stuff. You've written books about this. If we were smart right now, and it wasn't the Santa Claus program that was a $250 million bus that went down, was amazing. You'd think that you can't make this stuff up, but I guess you can. It happened in this country. But if you were to design a way to nurture public trust so that when the vaccines are ready, proved, and they have the data, so that we would get a high population level immunity, what can we do right now to foster trust?
I think right now, what we can do is be transparent with the process. And so I like that the FDA's vaccine advisory committee meeting was televised and anybody could log in and watch it. So they see people who are arguing about how this should work. There was one of the there was a press conference after that that meeting, and one of the reporters, I may have been an AP, an Associated Press reporter, said, boy, it looks so contentious. I mean, it looks everybody seems so skeptical about the vaccine. Yeah, that's what you want. You want everybody sitting around that table to be skeptical about this product. You want them to stand in the way in front of the American public and protect the American public. You want them to say, I will approve this vaccine when I would get it, when my parents would get it, when my children would get it. That's what you want to hear from those people, because that's the way it works at the ACIP. I was a voting member there too. And so I think once a vaccine is in hand and you can at least have some evidence that it works and is safe, then you have to be able to tell people what you know and what you don't know. You know that it's effective for X length of time. You know that it's X percentage effective. You know that it's safe in this many people for this length of time. Here's all the programs that are in place to continue to monitor safety, to continue to monitor for efficacy. Here's how we're going to continue to look at this. And then as it starts to roll out, 100,000 people have gotten it, 1 million people have gotten it, 10 million people have gotten it. And then you feel more and more safe. I mean, Maurice Hilleman, who I consider to be the father of modern vaccines, said it best, quote, "I never breathe a sigh of relief until the first 3 million doses are out there." That's always true. So you never really quite know.
Good.
You're always just trying to reduce uncertainty, but you never eliminate uncertainty. It's never when you know everything. You never know everything. The question is, when do you know enough to say the benefits of this likely, very likely outweigh its risk, and what are largely theoretical risks?
If you were to guess, just from the family of coronaviruses, and as good as we can get with vaccines, do you think we'll be able to go more than a year without people getting booster shots over time?
Yes. I think, I mean, I guess I should never, I'm willing to make any prediction as long as you don't hold me to it. But it's a single-stranded RNA virus. It certainly mutates, like any single-stranded RNA virus. But there's the measles single-stranded RNA virus and the flu single-stranded RNA virus. They're both also single-stranded RNA viruses. Flu mutates so much from one year to the next, or frankly, from 1 minute to the next. I mean, this is a virus that's constantly moving.
Oh, yeah,
The natural infection or immunization 1 year doesn't protect you the next year. I don't think that's going to be this virus. I mean, although you're right that there's been people who've gotten 2 infections and that there are genetic differences. The question is, and it hasn't really been answered yet, are there serotypic differences?
Right.
Are these genetic differences functional differences that make the virus a different serotype, or more virulent or less virulent, or more contagious or less contagious? And that's the important question. Not are there genetic differences? Of course, there's genetic differences.
Yeah, I mean, so far this has been such a slowly evolving virus, and there has, except for the one mutation, the D614G mutation, that has become dominant. And even then, it's debated whether or not it's truly, more infectious, that it's more, that it's more pathogenic. But I guess that point of a stable virus is good. But the issue is this concern that, it's hard enough to get, most of these programs have a booster shot initially, a second dose of the shot. And then every year to come back to maintain a population immunity. Plus, I think the other point that's worth noting is the vaccine is 60% efficacy versus 80 or 90%. The level of population that needs to be immunized is markedly different, right?
Yes, markedly different. Yes,
I mean,
there's a formula for this.
Yeah.
And if you had a vaccine, there's a few caveats to this, but if you had a vaccine that was, say, 75% effective, you would need to vaccinate about 2/3 the American population to stop spread. But there's other things going on. First of all, 75% effective probably doesn't mean 75% effective against shedding. But on the other hand, you do have some population immunity now. And also hopefully people will continue to wear masks despite the bad example that's being set by the administration.
Yeah, no kidding about that. Oh my gosh. And how you can not be anti-masking and pro-vaccine at the same time, just doesn't compute very well.
No, I was on service last week, and we take care of children who either have COVID-19 or suspected of having COVID-19. If you gave me the choice, when I walk into the room of someone with, say, suspected COVID-19, I have 3 choices. I can either stand 6 feet away, or if I don't stand 6 feet away and have to stand, say, within 2 feet of the person, I can either wear a mask or get a vaccine. It's, let's say, 75% effective. What would I choose? I choose standing 6 feet away first. I mean, social distancing is the most powerful. And secondly, I would choose the mask. I think the mask is worn correctly with the right mask. I'm not even talking about an N95 respirator. I'm just talking about a good surgical mask that's well fitting.
Sure.
That's going to be more than 75% effective. So the vaccine would be third, which is really the opposite of what the administration is currently pushing.
Yeah, that probably is one of the most important things to emphasize. In a little way, Redfield made those comments during a congressional testimony, and people said, "What? He'd rather have a mask than a vaccine?" But actually, you articulated that so much better, which isn't at all surprising to me, by the way. Okay, so what are your residual concerns at this time? We're going to see data from these trials, likely, a couple of them before year end. Fortunately, it's not going to be before the election in 8 days. It's going to be after. That got fixed, fortunately, by the FDA that seemed to develop a spine in the midst of all this. But perhaps we will see a couple of trials with readouts, and then we have a recommendation plan of phases, A1A and B, of who should get vaccinated, and kind of a stage process. What are your concerns about that going forward? Assuming that November, December, there's now it works.
I worry about the interim analysis with small numbers. That Pfizer and Moderna will say, look, we've had 35 cases of illness. And we're going to now report that. And we have 25 cases in the placebo group, 30 cases in the placebo group, and 4 or 5 or 10 in the vaccine group. I only hope that they don't submit for an EUA with small numbers. I really hope we get to 150, which is what the NIH active group that was put together by Francis Collins had asked for. I think that's minimum of what you need.
Right.
To at least feel comfortable that you have, so-called tight, confidence intervals for your efficacy, because you can't, obviously the worst thing you would do is put a vaccine out there that's unsafe. To put a vaccine out there that's ineffective, you are going to lose the trust of already what is a fragile confidence by the American public. And you don't get a second chance to make a first impression. And we need vaccines as our way out of this. And we can't screw that up. And I do worry that the pressure to get something out there quick with small numbers would put us, the FDA Vaccine Advisory Committee, in a position of having to say, you just don't have enough data yet, knowing that people are dying of this virus.
Well, you now have brought up the 2 points about this first trial, or the first couple of trials. Not only can they affect the rippling of all the programs of vaccines by declaring efficacy and making it placebo trial to maintain for people who are deemed at higher risk to stay on a placebo arm, but also this issue, which is incomplete ascertainment of what is really happening. That would be really a problem. Now, when these companies have their data, will your group be reassembled to adjudicate that?
Yes.
And that will be a public hearing as well, right?
The FDA, both FDA and CDC advisory committees are always open to the public.
Right. And they also, that data will, whatever company is, even though Pfizer is outside of the Operation Warp Speed, they'll be reviewed by NIH as well?
No, so here's how to work. We've already been asked, the FDA Vaccine Advisory Committee has already been asked to agree to 3 dates to meet in November and 3 dates to meet in December. Just in anticipation that there probably are going to be situations where we need to review data. But all the FDA does is the FDA essentially permits the companies to in the past sell the vaccine. Here it would be distribute the vaccine. But that's not the recommendation. I mean, it's the CDC that recommends vaccines through the Advisory Committee for Immunization Practices. So what the ACIP is going to do is they're going to independently review data and then make a recommendation. I mean, our hospital, I think, is going to be like many hospitals that will wait for a recommendation from the ACIP. And typically, insurance companies wait for the recommendation.
Right.
So I think the ACIP really is the key group. What worries me in all this is that after the hydroxychloroquine and convalescent plasma experience, it's not just that the public has gotten skittish about what's going on at the FDA. I think that professionals have. So now you have, at least 5 states that have said, we're going to form our own vaccine advisory committees. the National Medical Association, which is a group which represents African American physicians, have said the same thing. We're going to get our own group of vaccine advisors. We don't trust the government, and which is to say, we don't trust the FDA to protect us. If we've gotten to the point in America, we don't trust the FDA. We're in some trouble. I mean,
Yeah, well,
chaos.
this is yet another essential point, because earlier we talked about how can you promote trust, and what you've reviewed right from the top of our discussion today is that because of the precedent with the hydroxychloroquine that had no evidence basis, then the convalescent plasma, no evidence basis, and also in the midst of that, a widening EUA for remdesivir, which had very little to go with that, and now a full approval for remdesivir, which again has mixed results, including this recent WHO solidarity trial. You say, how can you approve it fully when you have a trial that negates the efficacy? So there's a trust lost in the FDA, but it seems as though, Paul, in recent weeks, the FDA has gotten stronger. That is, it stood up to the pressure of Trump and the administration and Azar and HHS. Is that a sign that things are turning in the right direction?
Yes, most definitely. I think when Commissioner Hahn stood up and said, no, we are getting 2 months of safety data after the last dose, which is a reasonable and fairly minimal requirement and typical, frankly, for vaccines, that clearly would have put the timeline beyond November 3rd, the election day, which clearly angered the administration. But he wrote it down, he submitted it in written form to us to consider, and the administration backed away. And I think it did make the administration angry. I think it may have made others who were higher up in HHS angry. But the fact of the matter is, Stephen Hahn stood up for the American public when he did that. So good for him. And as by turning to us, by turning to the FDA's Vaccine Advisory Committee, we're not government employees. I mean, we're academics. We'll give our opinion. We're used being in academic medicine. We're used to people ignoring our opinion all the time. But we can at least give an unvarnished, clear opinion about what we think about this. That's what we'll do. We'll be honest about what we think of this. And so if we do that, and that's a public meeting, and then there's any attempt to try and go around what we've done, I think it would just backfire.
Yeah, well, I think it's really great to hear your point on this. I mean, these are the most important clinical trials as far as I can tell, in history, because we're in such a horrible predicament. We've dug this humongous hole in terms of how we're going to dig out when we have 83,000 cases a day for a couple of days in a row, and it's still, on the rise. All the people that are, hospitalizations, and clearly now we're also seeing the deaths increase again. So we need a remedy. This is it, and we need to get it right. We've talked about all the things that could go off track if it's prematurely or, not properly given the green light. So this has been a great discussion. I hope that these other balkanization of reviews, like these states I'm in, California, New York, all these will start to get behind the FDA, that realize that you and your colleagues and these different external groups that are reviewing the data, as well as internal at FDA, they're doing it right. And I think we need to understand that this has gotten on course now in terms of the review process. And that's really great. We needed that because we didn't have that last time we met. It was all up in the air, what was going to happen, whether Trump was going to, waive his vaccine approval as an election tool ploy, So I'm feeling a lot better, and it's great to hear your views as well.
Thanks, Eric. Thanks for doing this. That was fun.
Well, Paul, this has been a great conversation again. I think we're going to have you back after your next review. There's no better way to understand what's going on than to get into your expertise, get your frank comments. And thank you so much for helping us at Medscape and our community to get us up to speed.
My pleasure. Thank you. Stay safe. All right.